So, what's the link between this and enzymes? Can you make a guess? What are the enzymes? What they do? What the slots are so slow? Why?
The enzyme can control the
Yeah, kind of. Yeah.
The enzyme analyzes the food into small pieces, like protein. When we mentioned that in the morning, the protein is divided or analyzed to small pieces, which is aminos.
Yeah, you are right. But the main part is that for enzymes, actually, what are the enzymes? Enzymes are biological catalysts. What are the catalysts? They increase the speed of everything. See, the point was that these losses, they can't actually go so fast. Why? Their metabolic is so slow. Their enzyme activity or converting something. What is the enzyme? Converting something to other things. Or helping for doing something. So we are going to see what are the enzymes. So, enzymes. I can see all names of the sessions here. So, protein acids, I guess this So, give me some examples of enzyme that you know. Yeah, pepsin enzyme. And some other enzymes. You know them? Exactly. Many of enzymes. In digestive system, specifically. Even in the market, do you have some detergent that they have enzyme? Have you ever seen those kind of advertisements? They say they have some detergents, they have enzyme, they help you killing the messes on your clothes. Yeah, you might have in your country. They are enzymes, they help you to digest lipids or some other things on your clothes. Yeah. And in industry, have you ever taken something to help you to digest something? Some people, they have a problem in digestive system. They can't digest many of some of these foods, and they receive some drugs. They have enzymes. What kind of food you are recommended after a meal? Do you have something in your country? They say eat this food after the fruit or something like that. After that meal, it helps you digest that
Any
enzyme.
kind of soda or vegetable.
No, they are not good.
Really?
Yeah.
They always advise you to take
That's not good. About the fruits, what kind of fruits? Have you ever kiwi? Orange. Orange is good for vitamin C, for absorbing iron. Because it helps for reducing that. How about the others?
Kiwi?
Kiwi, banana. Banana is not bad, but related to enzymes, banana has amylase. What happens to banana after, staying for a long time in a, outside?
Syria?
They have the taste. It tastes so sweet. What happens? Amylase works there. Amylase removes that glucose from amylose. Do you remember amylose? In starch. In starch, we had amylose. In banana, we have amylase enzyme on it. It digests that. What happens to kiwi if it stays for a long time outside? For the kiwi, what happens? It's a kind of soft. Why? It has enzymes on it. What kind of enzymes? Kiwi is good after a meal, that you eat a lot of meat. If you get some slices of kiwi, it helps for digesting that because it has digesting enzymes. If you put the kiwi on your meat, it can digest your meat in 20 minutes. Yeah, in some places like around the like kebab, they prepare it in one night before. But in some cases, when they are in a rush and they don't have time for putting that, just 20 minutes before the kebab, they start squeezing some of this kiwi juice on it. But if you put it more than 20 minutes, it can digest all meat because it has a lot of enzyme. If you look at that, it even digests themselves. What happens to kiwi after some days? You see that it's softened. It doesn't, because that enzyme works there, digests the kiwi. Even in coconut, we have specific enzymes. So, about the clinical importance, some of these enzymes are important. If you look at, have you tried some medical laboratory results in your home? Do you have laboratory results for your families at home? Do you have laboratory results for your family at home? If you have, next time, try to find some sugar tests, lipid tests, and some enzymes. That enzymes are important in clinics. Sometimes physicians, you might be at that point, you look at the enzyme levels. Some liver enzymes helps to get enough information about the liver. Because the concentration goes up. Or after heart attack, some enzymes go to the blood. So it is a clinical usage of that. And the best part. Using enzyme for skincare. It happens a lot. Some of these creams, they have some extractions of these fruits. They have a specific enzyme on it. Who's interested in making jelly? Have you ever tried that and putting some kiwi on it? So, does it work? When you put kiwi on it? Why? Kiwi digests that one. These jellies are gelatin, amino acid, proteins, and it digests that. How can you do it? Put your kiwi on freezer, kill the enzymes, or boil the kiwi and then put on it. The point. Yeah, because that's the enzyme. Enzymes are protein. If you freeze them or boil them, that proteins die, and activities die. Like this, they cut this amino acids and proteins. So, you are going to study this.
Yeah.
Is it good?
Yes. Yeah, it's very easy.
No, I'm kidding. I'm joking. You're not going to study this. But, imagine a lot of these reactions happens in one cell, just one cell. See, guys, see, all of these reactions happens in one cell, and we don't know they are doing their actual job, and we don't care about those things. All of those reactions happens by enzymes. Without enzymes, we face with the diseases, a lot of diseases. Some of them are congenital, some of them are related to vitamins because this enzyme needs vitamins for working. But all of these reactions happens by 6 group enzymes. 6 group enzymes. You are going to just learn the names of those things. 6 group of enzymes catalyze all of this reaction in the cells. In biochemistry, we have to know all of these things. And as a people, they want to work in metabolism because all diseases are related to metabolism. You eat a lot of something, accumulation of something, or you have, if you have a disease in one pathway, what happens? You will have accumulation of something. These are objectives for this part. You are going to learn the enzyme, what are the enzymes, what are their roles, and what are their classification, and in which condition they work properly. Definition of the enzyme. What are the enzymes?
Not each.
They are proteins. Yeah. What they do? What do they do?
Freeze the reaction.
Catalyze the speed of the reaction. Imagine without the enzyme, you should stay there like in this slot forever. For more than 10 years, one reaction happens. One glucose produces ATP. It doesn't produce that without the enzymes. So living without the enzyme is not imaginable. They are not altered or consumed during the reactions. They are intact. They can do a lot of things. Like, let's say I'm an enzyme here. My name is transferase. Transfer for putting this one from this side to the other side. This is called transfer. I'm not consumed. I'm just doing that one. I'm not altered during this process. And reusable. I'm here to transfer that, and again, if you have a lot of forms, I'm reusable. So, this is an enzyme, and this is a coenzyme part. Do you remember that coenzyme I mentioned above? Mainly, these coenzyme plus this protein part of the enzyme, this is the protein part, this is the non-protein part of that. If you remember that vitamins, they were not proteins. They are different compounds. But proteins always are protein. This part, without coenzyme part, is called apoenzyme. Plus this coenzyme is called holo. What does it mean, holo? Holo means total, complete enzyme. This is a coenzyme part here, this is a apoenzyme, and we call it holoenzyme. Mainly, you are not going to say holoenzyme. When you say enzyme, you mean enzyme plus coenzyme. Mainly, these coenzymes are vitamins. And in some cases, some metal ions, like iron, like copper, like zinc, we call it trace elements. Because they are in low, small amount, that is enough for working. That is enough for working of this enzyme. So, what are the cofactors? Cofactors are non-protein parts of the enzyme. They actually bound to the enzyme, and they are required and essential for these reactions. Type of the cofactors, if you remember, some of them are vitamins. See examples. Some of them are vitamins. Some others are metal ions, like zinc, copper, iron. And sometimes they need some other cations, like calcium, magnesium. They need for working that. Without that magnesium, they will not work properly. So because of that, those parts are essential for that enzymes. About the structure of the enzymes, how it works. Enzymes has a specific part which is called the active site. Like if this be a cutter and it's just supposed to cut this one, this part is active site. I can't actually cut this part by the other side. So the part of the enzyme, reaction happens there, it is called active site. See, this is the enzyme. This is the active site. What is this? Substrate. This enzyme, this is a substrate. If I cut this one, the substrate converted to product. So we have substrate, enzyme, the other side, enzyme, product. So enzyme, active site, substrate. Bonds to that, we will have, for example, here two products. Sometimes we have enzymes, it's going to connect these two together. We have here connect two, and the other side, our product is larger molecules, just one molecule. Combining two molecules together, it depends on the enzyme. Some enzymes are synthetic, some enzymes are catalytic enzymes. It depends on the pathway. We have two models for enzymes. Two models. Remember active site? We have active site. Some of this active site, let's say these ones, they are exactly same to the substrate. They are exactly same to substrate. Like this key we have here, lock and key. They are exactly fit together. For this model, we call it lock and key model. The active site of the enzyme is exactly same to the substrate. Substrate sit there and works the reaction. What is the limitation, or what are the limitations for these ones? Lock and key. What do you think about that? We have a specific active site, a specific substrate can bind to that. Is it good? Yeah, it is more specific. But what is the limitation? It is just for that one. It doesn't work with the other substrate. Do you like have a specific enzyme, its active site fit to that substrate? Depends on the substrate shape or different types of the substrate, it can fit that one. What do we call it? Induced fit model. Let's say this is an enzyme and I have a substrate like this. My enzyme can change the shape of the active site based on this substrate. For example, sometimes it's like this, sometimes like this. This active site shape can change based on this substrate. So what we call it? The shape induced this model. We call it induced fit model. For induced fit model enzyme, they can work in a different group of compounds. But for the lock and key model, they just specifically work. Which one of them is good?
The first one.
Both of them. Depends on your need. Sometimes you have a specific enzyme works on just glucose. Is it good? Yeah, it's good. Because you think at that time you are just going to work on glucose. Sometimes you say, I want to work on all sugars that they have 6 carbons. Not just glucose. For the glucose, you call it glucokinase, but for the 6 carbons, you call it hexokinase. What is the hexo? 6. Kinase is an enzyme for adding phosphate to that. So you say, yeah, it's good. If I have that option, it gives me more flexibility to work on different compounds. So because of that, we have 2 models. Some enzymes are more specific, some enzymes are fit-induced model. So both of them are good. And examples. The best example that I mentioned. some of them are more specific, like glucokinase or urease. What happens on urease reaction? Enzyme works on just urea. Let's find another example. Group of the compound. Group of the compound, like hexokinase. Hexokinase, all 6 carbons are done with reaction. How about the peptide bonds? We have a peptidase. It works on all peptides. Digest. If you consume peptide, meat is a peptide. Our enzyme can digest that one. So, based on that, this is a lock and key model. You can read it. I explained all of these things. What is that? This is a specific active site. This is fit to this model. If you compare, this compound is not fit. And but this one is more specific for that. How about the lock and? This is a lock and key model. This is the active site. This is the substrate specific to that. What about the fit model? The active site is like this, but substrate shape is like this. You have a diamond here, but the active site shape is a diamond. But this active site changes, shape and fit to that substrate. So, based on the specificity, this term is the most important term is specificity of in what level enzymes are specific. A single substrate, example, glucokinase, urea. When you have a specific names, it says it just works on it. How about this? A group of similar substrate. Similar substrate, like hexose, all hexose, all 6 carbons. Sugar, they have 6 carbons. Glucokinase, just glucose. In hexokinase, it means glucose, galactose, mannose, fructose, all of them, they have 6 carbons. And it works on them. And a particular type of bonds. What is the example for that? You know that? Bonds. What kind of bonds do you know? Between amino acids, between carbohydrates. Between carbohydrates is called glycoside. Glycoside. Glyco means sugar. Bonds between them is called glycoside. What is the enzyme for that? Glycosidase. Glycosidase. The name for the enzyme. The general name is that amylase. Amylase digests carbohydrates. All carbohydrates, polypeptides. How about another example for the proteins? Peptidase. All peptidase. They digest all peptides. They are specific for type of bond. You have a peptide bond, you have a peptidase. You have a carbohydrate bond, you have an amylase to digest that bonds. These are examples I explained in previous ones. You can find it here. So, what is the role of enzyme?
Increasing the metabolic reaction.
What?
Increase the speed of metabolic reaction.
Yes. It reduces the energy. Imagine without the enzyme. It showed not human does that. Imagine without the enzyme, we are going to see the enzyme reaction. It seems you need a lot of energy to do that reaction. And or time. It takes a lot of time to happen. But with the enzyme, what happens, it seems less energy for that because enzymes try to do that. So, just in one sentence, if I explain what does enzyme do, they reduce activation energy. They reduce activation of energy to catalyzing the reaction. last part of today's is those that said, do you remember all reactions? We have 6 types of the enzymes. What are the 6 types of enzymes? For some enzymes, for naming them, we have some traditional names for them. For example, for the sucrose, we add sucro. What is this?
Is.
Exactly. This ACE is an enzyme. For many things, for many enzymes, we have like this: glucokinase, urease. It actually is related to substrate mainly. Substrate, it works on it, the reaction does on it, and then ACE. Glucokinase, kinase is a kind of aminophosphate. Glucose is a substrate, adding the phosphate, and ACE is an enzyme. Some others are classified, are named based on the type of the reaction. For example, what does do this oxidase? What do you think?
Oxidation.
They oxidize. How about this reductase? Do we have reductase here? So how about the reductase? They reduce the reactions. They remove oxygen from them instead of adding them. Some others, actually, they use their common names actually for the digestive system, like pepsin and trypsin. It depends on the position that they hold for digestion. They have historical names. The classification. You need to learn these 6 names. What do you think about them? Can you make a guess about them? Look at them. What kind of reaction do you see in these groups?
Oxidation and reduction.
Sometimes oxidation, it depends on the enzyme. Sometimes reduction. They reduce or oxidize something. How about this one? For example, they transfer, this is a 2 substrate, enzyme transfer this part from this molecule to this one. At this part we will have this one without that group, and you will have that group on it, transferase. Transferase are most important because non-essential amino acids synthesize from essential amino acids. They help transfer amine group to carbohydrates, transferase, aminotransferases. They transfer amine group to carbohydrate to sugars and make amino acids. How about this?
Adding water.
Add water, break it down. Like carbohydrates or peptides, when you consume, you have to add water to that and break it down to units. How about the liases? I will ask at the end. Don't make a stop here. What's the isomerases? Do you know examples for them? Isomer. What are the isomers?
I know the meaning of isomers. It's similar to something.
Yeah, their formula is similar in writing form, but in the space, their positions are different, right? Like this one. When I'm like this, or I'm like this. They are isomers. Yeah, this is the examples of isomers. The best example, D and L, they are isomers. Glucose to galactose, they are isomers. Fructose to galactose, fructose to glucose, they are isomers. What is the usage of that? Imagine one mom wants to synthesize lactose. So what was the units of lactose? Do you remember that? Glucose and galactose. Glucose and galactose for lactose. So these units, if you don't consume galactose, what happens? Mom, they can't actually synthesize lactose? No, they can. They convert glucose to galactose, they themselves. But this is isomerase. If you consume galactose, what happens? You convert galactose to glucose in body with isomers. We have a specific pathway for glucose. So if you consume galactose, you don't have a specific pathway for that. You have some part, but mainly they are converted to glucose and glucose then to fructose. We have a specific pathway for that. So this convergence is a kind of done with isomerase. How about the ligases? Sometimes you need to connect more groups to this ones. Let's say not transferring. I'm gonna make a fuse of this. Add to this ones with energy. The only one that needs energy for reaction is this one. The rest, they don't need energy. See? They don't use ATP. They might transfer ATP, but putting money on pocket, but they don't consume that. Just except this one. This one consumes energy. So adding a group or fusing something to something. So, how about this one? Liases. It's a kind of complicated one between them. Among these groups, this one is a kind of more complicated because sometimes it adds water, remove water, making new compounds. It's a kind of enzyme. The difference between these hydrolases and lyases, what was the main definition for hydrolases? You add water, break something to units, not breaking it down. Let's say if I have a lot of sugars here, units. Adding water, remove the sugars. Units of the monosaccharides.
You're breaking the chemical bond.
Exactly. It breaks in hydrolases, you add the water to release units, just small units of that. But in lyases, you destroy that. That units destroyed, not large molecules, polymers. That the difference is that. So you are going to just learn the name of this. That's enough. On next slides, you will see examples, all of those explanations. What is this? What do you think here? Examples. Let's try your science on this part. Oxidation and reduction. What is the dehydrogenase? It's a kind of oxidation and reduction. What is this? Transaminase. It's a kind of transferase. What is this? Adding water to breaking this one. It's called hydrolase. It's so easy. This is the lyases. Add water, but changing these molecules, not removing units I mentioned. See? Changing that, rearranging that, not removing part of that. What is this isomerases? You converted L-alanine into what? D-alanine. And this is the isomer for glucose to fructose. Converted that, and finally, you have a molecule here. You want to add to this amine groups to this new bonds with consuming what? ATP. Just one group consuming ATP. The ligase. All of things, these two slides are explanation of those ones. Everything that they explained you can find here. And these two. So, the last parts of the enzymes are environments that affect enzyme activity. One of them was cofactors. In deficiency of vitamins, enzyme will not work. So you should get enough vitamins. How about the environmental? Environmental condition is related to pH and temperature. In high temperature, enzymes are protein, they will die. Because of that, we can tolerate more than 45 temperature. In 42, that is so high fever, and we can tolerate that. In 45, we will die, because many of these proteins will die. And enzyme inhibitor. The most interesting part is that controlling enzyme activity. With what? Drugs. Many of the drugs or toxins they control these enzymes. They are called enzyme inhibitors. They are not necessarily bad things. Sometimes we want to control our enzyme activity with drugs, like anti-blood pressure drugs or painkillers. They actually stop enzyme activity. Some toxins also kill the enzyme. Or antibiotics. Antibiotics stop bacterial enzymatic system for synthesizing membrane or synthesizing proteins. So studying the enzyme is the most interesting subject because all reactions happen with enzyme and you can control it with inhibiting it, targeting a specific drug. I'm not going to explain one by one these ones. I explained this environmental for the temperature. See, the optimum temperature is around the 37. In low, this is the turnover number or activity of the enzymes. It is low when the temperature is low. When the temperature goes up, it is going up, and the optimum is here. If you increase the temperature more than 40, 45, see, in this part, you don't have enzyme activity. This is the enzyme activity, it goes down. And also about the pH. Some enzymes work on acidic pH, like penicillin in our stomach. Works on this pH. If the pH changes, it doesn't work. It doesn't digest because it works on this pH. And for the amylase, it works on a neutral or on the acidic. How about the trypsin? It works on basic. If you don't have enough basic condition, this trypsin will not work. And for the drugs, last things, we have two kinds of drugs or inhibition of enzyme. Some of them are reversible, some others are irreversible. It depends on the treatment policy. Mainly we prefer these reversible ones because like ibuprofen. You can see ibuprofen or gelofin? Ibuprofen. Yeah, it's a painkiller. It is a competitive and reversible. For reversible, we have two kinds of inhibition. They are competing with the active site, they compete with the substrate, they sync to the substrate with the active site binding, inhibit enzyme. Let's say my work is that transferring to this side. If something looks like this and occupy my hand, what happens? It inhibits my activity. In competitive ones, the substrate same to drugs, something like this. I don't know, something, let's say we have something here, make my hands busy and I can't actually transfer this. It's a kind of competitive inhibitor. Many of drugs are like that. Many of drugs, like ibuprofen. But for the irreversible ones, how about the aspirin? Have you heard about aspirin?
Yeah.
What is that?
To
Reduce the pain. Chest pain. They work in the same enzyme, but ibuprofen and gelofin are reversible and competitive part of this reversible, but aspirin is irreversible. When it bonds to that, they destroy the enzyme. It takes 3 days to replace that enzyme. But for this one, after 8 hours, the enzyme works properly. Just reduce the activity of that in this form. There are some examples here, and one example that this is aspirin bounds to this specific place with ibuprofen and gelofin. This is example for antibiotic working. Antibiotic stops bacterial enzymes activity. So, and it helps as an antibiotic. And the final things, that's the final slide that we have. A general overview of factors influencing enzyme activity. Substrate. I am an enzyme. If I don't have a substrate, what happens? I can't work in high speed. I should have substrate to work. If I have more product, if I have let's say this is my product here. If I am